Hi everyone, and welcome to our video on the mechanism of enzyme action. What we're going to be looking at in this particular video is our specification reference 2.1.4c, which is quite simply the mechanism of enzyme action. So we're going to have a little look at things like the active site, lock and key hypothesis, etc. Now, the first thing we're going to do then is consider the actual key parts, if you like, of an enzyme molecule. Now, I'm sure that you remember from your GCSE studies that your teacher probably drew something along the lines of this little kind of shape, and we basically had kind of like a little Pac-Man. Now, what we will find is we have this overall shape because it is a protein. Now, proteins, if we remember, have that tertiary structure, which is their three-dimensional shape from our earlier work on proteins. And that gives us this key region right here. And that is called the active site. Now, the active site is going to act as what we refer to as the lock in this lock and key hypothesis. And basically that means that when we're looking at the substrate, so the thing it's going to obviously have a reaction with, then that is going to be the key. So what we find, the active site and the substrate have complementary shapes, and therefore when they actually combine, it's this perfect fit, so to speak. Hence, lock and key hypothesis. Your key fits the lock. Now, in terms of the additional detail here, what we need to remember are some of those kind of key phrases, if you like. So we've got our enzyme, obviously the protein that has that specific three-dimensional shape, and then we've got our substrate. So this is the bit that's going to actually fit into the active site. Now, both the enzyme and the substrate have kinetic energy because they are going to be moving. And what we find is because they're both moving with this kinetic energy, then there's a chance of a collision. If they collide, then we have the possibility of forming this enzyme substrate complex. And that is quite simply the substrate molecule fitting into the active site of the enzyme. So once that happens, we now have this thing called the enzyme substrate complex. Depending on what kind of reaction this enzyme is catalyzing, that substrate is either going to be broken down or it's going to be combined, so joining those two reactants together in order to make the product. At that point, we now have the enzyme product complex because the product is still held within the active site of the enzyme. Last thing that has to happen, the product gets released. So this is just giving us that very basic overview of the lock and key hypothesis. Enzyme substrate moving with their kinetic energy, they collide, form the enzyme substrate complex, they catalyze the reaction to form the enzyme product complex, and then the product is released. We now need to have a look at a new hypothesis. So this is one that you will only hear about at A level. Now, the induced fit hypothesis is going to explain how the enzyme substrate complex is actually stabilized. And what we mean by that is that when the substrate fits into the active site, then the induced fit hypothesis tells us that the active site is going to change its shape to literally mold itself around the substrate molecule itself. And the reason that this is going to occur is because we're going to get very slight changes in the shape of the side chains of those amino acids that make up the active site. So realistically, what we've got in our enzyme is obviously a polypeptide. The polypeptide is going to be made of all those amino acids joined together. Each amino acid has its own little side chain, that R group. Now, if we're going to make these slight changes to the shape of the side chains, that's going to allow the active site to mold itself around the substrate molecule itself. When we actually consider how that substrate is bound to the active site, when it is in this enzyme substrate complex, we need to remember the names of four bonds or interactions. The first one is our old friend, the hydrogen bond. Second one, ionic attractions, which are obviously going to form between oppositely charged parts of a molecule or molecules. Third one are van der Waals forces. Now these are just little weak interactions basically. And then we've got hydrophobic interactions as well. So what we find is that these 4 different interactions and bonds that we could find are then going to hold that substrate in the active site when we have it as the enzyme substrate complex. Once the enzyme has done its thing and it's catalyzed that reaction, then we've obviously formed new product. Now that product that we have now created is going to be a slightly different shape to the original substrate. Where it used to have this absolute perfect fit, it doesn't anymore. So we've got a different shape as the result of the reaction that the enzyme has catalyzed, and so we will now see the product detaching from the active site. The last thing we really need to know about in this particular video is activation energy. And again, this should be familiar to us from GCSE. When we talk about activation energy, we are literally talking about the energy needed to start a reaction. And when we use an enzyme in one of these biological reactions, what it's doing is it lowers the activation energy. So what we mean by that is it's going to reduce the amount of energy required to actually start that reaction. Hence it catalyzes it. How does it achieve this? Well, what it's actually going to do is it's going to bring the substrate molecules close enough to actually react without the need for the high temperatures. So pretty much if you imagine the little enzyme almost acting like something that's drawing these molecules nice and close together, so that it's almost a forced introduction in some ways, and that means that they're going to be close enough to react, but we haven't needed that high temperature and therefore the larger amount of energy to bring that about. And you can see in the little graph at the bottom there how this looks in one of the reaction profiles. So our red line there is going to be without the enzyme. So what we can see is we do have this significant activation energy. And then our little black line is going to be with the enzyme. So we can see that it has a much lower activation energy with the enzyme than without, which means less energy goes in to actually catalyze that reaction. As always, don't forget to subscribe to the channel so you can see when another video is uploaded, and head on over to the A-level website so that you can get hold of any other resources that may be of use to you in your A-level studies.